What up, I'm H-Minute Peeps. This is Taiwan Hubbard, and we're here to do a research update video for you guys. This particular article was published May 26, 2001, and the article is titled Mechanism of Hydrogen on Cervical Cancer Suppression Revealed by High-Throughput RNA Sequencing. So I wanted to highlight this study for a few reasons. Number one, I wanted to highlight it because it is recent. It's a recent publication. And then number two, it's an article on cancer. They've been doing a lot of different studies and investigating hydrogen in cancer. And then number three is over cervical cancer. So I only have a few sources for hydrogen therapy in cervical cancer. And this one's actually giving us some potential mechanisms behind how hydrogen is able to suppress cervical cancer. And so I wanted to go through the abstract and give you some basic information about the article, and then we can go through the discussion and then look at some of the actual highlights of this article, which I think are pretty interesting. There's one that is quite peculiar that I think will be a fun one to talk about. So let's go ahead and begin. All right, so in this article or this study, they are investigating in vitro and in vivo. So they're looking at cell cultures, and they're also going to be doing a xenograft of a cell culture and putting it into mice and looking how hydrogen could be affecting tumor growth within these mice. So the administration method that they're actually looking at is hydrogen inhalation. They use two different concentrations, a 33% concentration and a 66.7% concentration of hydrogen, so a higher concentration. And then they actually record the tumor growth within the mice every 3 days, and they go out to 34 days when they actually take their evaluation based on the hydrogen therapy group and the control. So that's the basic information of the study. Let's go ahead and look at the discussion and see some of the key highlights about the study. So to begin, I want to kind of discuss some of the highlights of just basic information that they talk about with cervical cancer. There's some information I didn't know about, so I figured it'd be worthwhile to share. So here's a quote. It says, cervical cancer is the fourth most common type of cancer and fourth leading cause of cancer-associated mortality among women worldwide. In addition, cervical cancer is the second leading cause of cancer-associated mortality in women aged 20 to 39 in the United States, and it's estimated that there may be 13,800 new cases and 4,290 mortalities in the year 2020. So, number one, cervical cancer is quite the problem. It is a disease model that they are looking at and trying to find other treatments that could be a useful strategy against cervical cancer. Furthermore, it goes on to say this about cervical cancer. It says that 70% of cervical cancer is caused by HPV 16 and 18 infections. And so it's been associated that, HPV, by the way, has been HPV 16 and 18 are primarily associated with squamous intraepithelial lesions, which may progress to invasive squamous cell sarcoma, such as cervical cancer. So, it is quite the problem, and they're looking at how hydrogen can potentially aid as a novel therapy for this particular disease model. So let's look at some of the highlights with hydrogen. First thing I wanted to go over is that they actually talk about how in the in vitro part, or in the cell culture part, that the 33.3% hydrogen gas actually caused significant or it significantly elevated apoptosis rate and reduced the cell proliferation and oxidative stress in the cancer cells. And then it says this, that the 66.7 though percent increased the apoptosis rate and reduce the cell proliferation even more so. So it looks to be that hydrogen has a dose dependent effect on these cervical cancer cells, and a higher dose led to a greater response. This is also a trend that is being seen throughout other disease models, and other studies that are being conducted, that certain disease models with hydrogen seem to be dose dependent, and a higher dose appears to be more effective than a lower dose. Now, there's way more studies that need to be done on dosing with hydrogen, but at least for this article in particular, the higher concentration was more effective, not only for the in vitro, but also for the actual the mice themselves that had the tumors in it. So it says here that hydrogen was able to selectively have a apoptotic effect on the cancer cells, but they also investigated hydrogen's effects on skin cells. I think they're called keratinocytes, and they noticed that hydrogen did not have a suppression effect on those skin cells, but it did have it for the actual cancer cells themselves. A H2 concentration of 33.3% significantly increased cell apoptosis and reduced the cell proliferation of cancer cells, but not those of non-tumor cat cells, or basically skin cells. Furthermore, a high concentration of H2 inhibited cancer cell proliferation and promoted cell apoptosis. This data reveals that H2 has a dose-dependent role on cervical cancer suppression. Another interesting thing that they observed and noted in the study is that 66.6 or 66.7% H2 has similar cell apoptosis or cell death rate of that of a known and approved chemotherapy drug. And so I'll go ahead and read the quote for you. It says, importantly, the increased cancer cell apoptotic rate induced by 5 micrograms per liter cisplatin treatment is similar to the effects of 66.7% H2 treatment, suggesting that H2 therapy may be a novel therapy for cervical cancer with less toxicity and efficacy comparable to chemotherapy. That's pretty awesome that they observed in the study that hydrogen had the similar rate of cellular death as cisplatin, or which is a known chemotherapy drug. Now this is where it gets into something quite interesting. I will say basically what they observed, and then I'll come back to this point a little later. But it says here, unexpectedly, H2 intervention reduced oxidative stress levels, including decreasing ROS concentrations and increasing superoxide dismutase levels, which appear to have the opposite effect on tumor cell suppression. It's interesting that they noticed that hydrogen was able to, hydrogen lowered oxidative stress within the cancer cells, which would be contrary to popular thought of what we want. We want to increase oxidative stress to a really high level, to a point of killing the cancer cells. That's what we want. So we'll come back to that point, but they basically provide an explanation of maybe why hydrogen had this effect. But it's interesting. It's interesting nonetheless. It says here that they noted that hydrogen had tumor inhibitory effects on the actual tumor-bearing mice. They noted that after 34 days, they compared hydrogen treatment to the control and noticed that it had decreased the tumor in weight and size and cell proliferation, and increase tumor cell apoptosis. And so hydrogen inhalation was able to do this and have these effects, or having these effects on these mice within 34 days. So now we get into the mechanisms. So they go through a particular. They use, like the title said, they use RNA sequencing to be able to identify some of the possible mechanism how hydrogen is able to induce these effects. And it looks like it's doing it in a number of ways. It seems like this is a trend with hydrogen. It looks to be having a multitude of effects, possible effects, not just one or two. So they elucidate a few possible mechanisms that it is actually inducing in these cancer cells. One is with non-coded RNAs. So this would be microRNAs, long non-coded chain RNAs, circular RNAs. And so they said here that they noted that the present study revealed that 11 circular RNAs, 2 long non-coded RNAs, and 12 microRNAs were upregulated, and 16 circular RNAs and 6 long non-coded RNAs and 2 microRNAs were downregulated by H2 in the cancer cells. They suggest here also that this effect on these microRNAs or these RNAs may account for the tumor inhibitory role of H2 on cervical cancer cells. And may be a specific mechanism behind how hydrogen has cancer suppression effects. And so that's just one, as hydrogen's influencing microRNAs. They actually have an important role when it comes to cancer. They actually state here that these microRNAs have been associated with regulation of cell proliferation, apoptosis, and changes in their expression may be responsible for proliferative diseases, including cancer. So they have a very important role when it comes to cancer. Another mechanism that they actually look at is hydrogen was able to downregulate NF-kappa B P65, which is a subunit of NF-kappa B. This is a molecular pathway and protein that is normally seen highly in the inflammatory response. But it has been associated with cancer. It says here, it has been reported that NF-kappa B65 is a molecular linchpin that has been linked to chronic infections and inflammation with elevated risk of cancer. NF-kappa B persistently activated in multiple cancer types exert pro-tumorigenic function. So cancer cells like NF-kappa B, and they induce it and elevate the levels of it because it helps them to survive. Well, they noted that hydrogen was able to downregulate this protein within cancer cells, within the actual cervical cancer cells. It says here that H2 intervention in cancer cells and tumors were demonstrated by reducing both NF-kappa B 65 protein and microRNA levels in the RNA sequencing and molecular biology experiment in the present study, suggesting a decreased inflammatory response and tumor growth. So it is reducing the inflammatory response around the tumors as well as reducing tumor growth. That's pretty interesting. It also says here another possible mechanism behind how hydrogen is doing this is by downregulating what's called hypoxia-inducible factors. But these hypoxia-inducible factors are, we identified 3 of them. They are 1 alpha, 2 alpha, and 3 alpha. But basically, hypoxia is a lack of oxygen. And so this has been characterized around cancer microenvironments that they have this hypoxia environment. And these particular proteins help cancer to survive. And so it says here, talking about the hypoxia inducible factor 1 alpha, the overexpression of this protein is strongly associated with the elevated metastasis and mortality in numerous human cancer types, including cervical cancer. Upregulation of hypoxia inducible factor 1 alpha in these cancer types results in a series of regulatory processes leading to tumor progression, including migration, invasion, angiogenesis, glucose metabolism, and pH regulation. So the upregulation of this particular protein allows, or at least is an aid for cancer to maintain all of those characteristics in which I just mentioned. So it says here, though, that hydrogen was able to downregulate this protein. It says here in this quote, it says, the present study demonstrated that hypoxia-inducible factor 1 alpha protein levels were reduced in H2-treated cancer cells and tumors, which is important for elucidating the potential mechanisms for H2 gas on cervical cancer. Notably, now back to my point about the reduction of oxidative stress when it comes to cancer cells. It looked like this should be that should be a problem. You don't want to reduce the oxidative stress. It says here, though, notably, the reduction of oxidative stress observed in the cancer cells may contradict the anti-tumor effect of H2. This is what I was just mentioning. It says, however, ROS or oxidative stress reactive oxygen species are essential in the stabilization of hypoxia-inducible factors that maintain their transcription of genes involved in tumor development. This downward trend of oxidative stress may be considered an anti-tumor effect in general. So it's saying that hydrogen was downregulating this particular protein that is used by cancer, and one of the things they observed was that they noticed a reduction of oxidative stress within the cancer cells, although they were dying. So this seems to contradict, but this reduction in oxidative stress could have been how hydrogen is actually downregulating, or at least possibly downregulating hypoxia-inducible factor alpha. So it almost seems like it's something new every time I read one of these studies with hydrogen that is inducing these effects in some of the most non-normative ways, especially compared to other therapies. And so that is something that I have not seen in other hydrogen studies. I mean, there are a lot of hydrogen studies. I've read a lot of them, but they seem like there's a new one that comes out every day. And there's a new, there's a new angle or twist in how hydrogen is actually being able to induce these effects that does not seem normative. So furthermore, it says here that they believe that it can be theorized that hydrogen is also, theorized that hydrogen also may be regulating the HPV-18 genome within the cancer cells, thereby exerting tumor suppressant effects. We need more research, but that is pretty remarkable to think about that hydrogen could be regulating the genome of the cancer cells, causing apoptosis, and regulating, because these cells are infected with a virus, regulating the genome of the virus within the cells, causing tumor suppressant effects. So that's another possible way that we need more research on it, obviously, but that's another possible way of how hydrogen could be inducing these effects. So this is the main highlights from the study. I'll read the conclusion for you guys and wrap this on up. In conclusion, the present study suggests a novel H2-induced tumor suppression towards hypoxia-inducible factor 1 alpha and NF-kappa B. Inhibition of hypoxia-inducible factor 1 alpha and NF-kappa B, reduction in cervical cancer cell proliferation, oxidative stress, and decrease tumor growth, which makes H2 a potential target in the treatment of cervical cancer. So, hydrogen being able to downregulate these particular proteins had a anti-cancer effect, and anti-tumor effect, that comparable to a known chemotherapy drug. So that's pretty cool. It seems like hydrogen has the right trend, has a really good trend and trajectory right now. Through the literature, just continues to get better. We continue to find out more possible mechanisms, and we'll be here to bring you more updates about hydrogen and different disease models and recent studies. And so we want to make sure we get back into a good rhythm with these. COVID season is COVID season for everybody. So we haven't been able to be as consistent as we like to with research updates, but we want to get back on that trend so you guys can get to hear more about these amazing studies that are being published. So we'll catch you next time. Deuces.